Hello, good morning students. We have studied about the process of photosynthesis, the definition of photosynthesis, adaptation, then factors affecting factors which is external as well as internal factors, then various adaptation in plants for to facilitate or to increase or to reduce photosynthesis process, then importance of photosynthesis for plants as well as for other organisms. Then we have studied about the carbon cycle. Um, all the factors, all the terms related to this we have already discussed. But all the terms, all the facts which we are saying is correct or not, it requires an experimental proof. So today's video is all about experimental proof to show that we are saying that CO2 is necessary, we are saying that sunlight is necessary, chlorophyll is necessary, and oxygen is a byproduct. So what are the experiments or what are the experimental proof for these facts? So today we will study about the experiments to show that whatever we have studied is correct for the process and which is experimentally proved. Then for this we require a discharged plant. So what is the process of discharging? First, discharged plant is in discharging we take when we take a leaf of a plant which is kept for 2 to 3 days in a dark room. Keeping a plant in a dark room makes it discharged. Discharged because the starch, the component which is from glucose to starch conversion, the starch is utilized by the plant during this process when it has not photosynthesized as it is kept in a dark room. So lack of photosynthesis resulted in discharging of the leaf when it is kept in dark room for 2 to 3 days. After that, there is a process to remove the chlorophyll content or to destroy the cells to stop the enzymatic action. For this, the process is the leaf sample is kept in boiling water. Keeping for a minute, keeping in boiling water destroys the cell or kills the cell, because if the cell is living, then the enzymes would be activated, would be in an active state, and further reaction would proceed. So for this we are keeping the leaf sample in a boiling water for a few minutes. Then the leaf sample is again boiled in a methylated spirit. Methylated spirit, it catches fire very easily, or it is inflammable. So for this we to avoid this catching fire by the methylated spirit, we boil the leaf sample in methylated spirit in a water bath. And now what is the water bath? For this we will take a sample in a beaker, we will take water up to a level, and inside the beaker in a test tube we will take a leaf sample. Then the test tube with leaf sample and methylated spirit is kept in the boiling water. Water is boiled on the over a spirit lamp. When it is kept over a spirit lamp, water is boiled, and with the temperature of water, hot water, this methylated spirit also gets heated. So this heating of heating in a methylated spirit removes the chlorophyll content, but the leaf becomes pale yellow in color and brittle. To make it soften, we are again keeping the leaf sample in hot water. The leaf becomes softened, and after that we are removing it in a petri dish and pouring a few drops of iodine solution on the leaf. If the color changes to bluish black, it shows that starch is present. If the color turns to brown, it means starch is absent. So to prove that generally if the proper process is followed, the color occurs brown in color, because the leaf lacks chlorophyll and it is a discharged leaf. Now for the experiment, first experiment is to show that chlorophyll is a necessary pigment for the process to take place. For this we are taking a variegated leaf of coleus or croton or geranium. The variegated leaf means the leaf contains green area in patches. Not the complete leaf is not green in color. Patchy appearance of green color here. In this I have shown this area for green. This area is marked for green. And this is colorless area, or colorless or not colorless, white. It appears white. So for this experiment, when we have kept the leaf for 2 to 3 days in dark room, the leaf is discharged, and when we have tested it for with the iodine solution, the area where the leaf is green in color shows it turns bluish black to show the presence of starch, as the photosynthesis takes place only in the green areas where chlorophyll is present. Green coloration is because of chlorophyll. So green areas turn bluish black, while the area which is non-green, it appears brown. No starch is produced in this area. So this experiment proves that chlorophyll is a necessary pigment for photosynthesis. Next experiment is to show that sunlight is necessary for the plant. Sunlight is necessary for the plant. We have taken a leaf sample, discharged leaf sample. Now discharged, you all already know how the leaves are discharged and why it is discharged. Then it is covered with a black strip in this area, and with a cut design. Star design is cut on the black sample so that this area would get light. So all the area here which is made check in the cross check here, this is it shows the green portions while this is black strip. Now when this leaf is tested for starch, the area which is green in color or which is uncovered shows the presence of starch after iodine test, and the area which is covered with the black strip shows the absence of starch in it. It proves that sunlight, when sunlight is falling on the areas where sunlight is available, or sunlight falls on the area shows presence of starch, photosynthesis takes place, while the black portions or the portions covered with black strip due to unavailability of sunlight shows non-photosynthesis or no starch zones or brown coloration. So this area shows bluish black, and this black strip area, it becomes brown in color to show no starch zone. Next, the questions related to these two experiments. First one, why we are taking a discharged leaf? What is the process of discharging? Why the plant is boiled? What is the boiled in? Why the plant is why the leaf is boiled in is kept in a boiling water? Or killing of cells is to destroy enzymes. This one. So this you have to mention that boiling water is to kill the cells to stop enzymatic action. Then the question related to what is the color for the to detect the presence of starch and to detect the absence of starch. So bluish black color for starch zone and black color brown for non-starch areas. Here for this experiment, what is the aim of the experiment? For every experiment, aim of the experiment is very important, because it is asked what does the experiment shows. So the experiment shows that sunlight is necessary or chlorophyll is necessary for the plant. Then the basic question is discharging of the leaves. and about these processes. Then the next experiment is to show that CO2 is necessary for plants, or CO2 is necessary for the process of photosynthesis. So in this we have taken a conical flask. Inside the conical flask, potassium hydroxide is placed and a leaf is placed in such a manner that half of the portion of the leaf is outside the conical flask, which receives the sunlight, and here half of the portion is inside the conical flask, where no CO2 is present because potassium hydroxide absorbs carbon dioxide. So this, after some time, the leaf is taken out, and in this case also discharged leaf is taken, and then it is placed like this and then kept for sunlight, kept in sunlight. All these leaves are kept in sunlight for experiment. 2-3 hours we are keeping it in sunlight. After discharging, it is kept for 2-3 hours for in sunlight for the process of photosynthesis to take place. Here in this experiment, when we have tested this leaf, the leaf which is for the sample for the experiment, the leaf which is inside the conical flask shows no starch after iodine test, while the leaf which is outside the conical flask shows presence of starch, as it is receiving the sunlight as well as the CO2 is available. Sunlight is available to this also, because the conical flask is a glass flask. So sunlight is available to this one also, but CO2 is not available. So here the portion of leaf where CO2 is available shows bluish black in coloration. Starch is present, it means photosynthesis takes place, while the leaf which is inside the conical flask shows no starch area, or this one, absence of starch. Now this question related to this, first, aim of the experiment. Second thing, what is the role of KOH? Role of KOH is to absorb CO2. Third question, it is what are the observations in the leaf, which part shows the presence of starch or which part shows the absence of starch. So these 3 questions related to this experiment. Next, one more experiment. All these are the requirements for the process to take place. Then after that, what we have studied that oxygen is released out. So to show that oxygen is released out, we have to show with an experimental proof. So for this, one more experiment. In a beaker containing water, pond water, we have taken a short stem conical flask, and inside the conical flask a hydrilla plant is kept. Hydrilla. Here. Now over this stem a test tube is placed. Test tube is placed over the stem. Now this arrangement, in a beaker, pond water is taken. In the pond water, the stem of hydrilla is kept in a conical flask, inverted conical flask, and this is kept in sunlight for a few hours. After keeping in sunlight for a few hours, what we will observe is that air bubbles comes out, and this in the test, the water level in the test tube falls down because the oxygen released during the process displaces the water downwards and displaces the water. If this is tested with a burning sphincter, then it burns with a bright glow, showing that oxygen is released during this process. So this experiment is to prove to prove that O2 is released during photosynthesis. Hydrilla plant in a inverted conical flask. It is kept for 2 to 3 hours in bright sunlight and the process takes place. Thank you for today's session.